Hello everyone. I need to go back and review some of the details and then add some detail to your background for your public service announcement poster about a genetic disorder. So before cells are going to divide, they condense their DNA into a structure referred to as a chromosome. So this entire thing is a chromosome. And chromosomes have a top section or a lobe and a bottom section or a lobe. And these sections or lobes are called chromatids. Again, be sure and add these to your notes. It's going to be the next to last section that we're going to add to your notebook. And then these two sections or these two lobes are held together by a protein ball called a centromere. So here's your chromosome that forms right before the cell's going to divide by either mitosis or meiosis, which is what we'll talk about next week. And then the two chromatids are held together by a centromere. And then don't forget that this chromosome is made of DNA, condensed DNA, DNA that's super coiled to make this structure called a chromosome. Well, now to connect this to genetics, we've been talking about things like big R and little r, a dominant allele and a recessive allele. And we've been calling this your genotype, what you carry in your genes. Well, then we also know that a gene is a section of DNA. So this chromosome contains your genes. This chromosome contains the different alleles that you inherited from your biological parents. So what we need to do is we need to connect your knowledge here between a couple of different units. So here is a chromosome. And this chromosome is made of thousands of different sections of DNA called genes. So when we write big R, little r, what that means is we carry two chromosomes, what we call a homologous pair. And let's make this, I don't know, chromosome number 12. Fine. So here is your homologous pair number 12. You receive one of these chromosomes from your biological mother and the other chromosome from your biological father. And these genes have specific placements or locations. They're called a locus. Here is one locus, here is another locus, or together they are called loci. So this is the locus for some characteristic. So from your, let's say your biological mother, you received a chromosome that has the dominant R on it. And then from your biological father, you received the chromosome that has your recessive R on it. Notice how they are located on the same position of each chromosome. However, the gene that they express is different. So then what happens is this gene, okay, don't forget that you have a gene, and that gene is read by transcription, making messenger RNA. That messenger RNA is then translated, transcription, translation, remember? So this gene, this one is expressed because it's dominant. There's a whole nother layer of biochemistry here we're not talking about, of how is this one dominant and this one is recessive. You will talk about that if you're a biology major when you're a junior in college in a class called biochemistry. You're going to love it. So here is your dominant gene. That dominant gene gets read, transcription, produces messenger RNA. That messenger RNA is then read by the ribosome, linking amino acids to make a protein. When that cell expresses that protein, what we see is your physical appearance, your phenotype. Thus, your genotype leads to your phenotype. That's why transcription and translation was so important. Okay, so chromosomes. Chromosomes are made of these two pieces, these two sections, called chromatids. And we need to talk about those chromatids because there are some instructions in your genetic poster that I need to be clear on. So when you look at a chromosome, the top chromatid and the bottom chromatid are never the same size, okay? The top chromatid is always smaller. But if I put an S on that, the top chromatid is always smaller, and the lower chromatid is always larger, always, okay? So the first scientists that discovered this were French. And so since they were French, they used the French word for small. They used the term petite. It's a French term. Well, since this is the petite chromatid, we refer to it as the P chromatid. Why? Because it's petite. It's small. It's small for French. Excuse me, it's French for small, right? Whereas this chromatid is larger, so then you would assume that they would then use the word for large in French. Nope. They followed the alphabet, and HIJKLMNOP, what letter comes after P? Q. I think it would have been cool if they'd used the letter for longer or larger in French, but I wasn't on the committee. So we refer to the top chromatid as the P chromatid. So this is the P chromatid. And the longer, lower chromatid as the Q chromatid. P's and Q's. P's always on top, okay? So then to go back to our earlier chromosome that we drew as number 12, and I have this habit, as I've actually shown you right there, I don't always draw the chromosomes the correct size, that this should actually be smaller. And then we put the R here, and we put the R here. And you will see how this is then labeled. So if this gene is located here, what we would do is we would say this gene is located on chromosome number 12, right? Because it's the 12th chromosome. It is then located on the P chromatid. And then if you stain chromosomes and put them under ultraviolet light, there are these bands that appear. And you can Google this. Google stained chromosomes, and you will see how the chromosomes have very, very distinct banding patterns, okay? Well, if you look at what I've drawn here, here's 1, 2, 3, 4. Our R allele right here is the 4th band down. So it's the 4th band on the P chromatid on the 12th chromosome. Well, how do we write that in genetics? On the 12th chromosome, on the P chromatid, and it's the 4th band. So that is how you would identify the allele dominant R or recessive R, whatever phenotype that expresses for. Okay? So on your handout, when it lists the number, the letter, and the number, that's what it means. This is the chromosome number. This is the chromatid, the upper or the lower chromatid. And then this is the band on the chromosome itself. So when you look up your genetic disorders, they will be located on a specific chromosome, a specific chromatid, and a specific band. Good luck, and let me know if you have any questions. I'll talk to you soon.